Well, what we're going to do today is look at a couple more and two, possibly three more major developmentally important signaling pathways. Let's see if I can get a little bit better view here. Okay, that shouldn't be too bad. All right, okay, good. All right, and those pathways are the RAS MAPK pathway, the TGF or transforming growth factor beta pathway, and we'll see if we can move on beyond that. Now, we left off on Thursday describing these growth factors, these numerous families of small secreted proteins, many which activate this particular pathway. Not all of them do, by the way, but many of them do. And they're widely used in things like tissue maintenance, wound repair, tissue repair, organ repair, and all kinds of developmental processes. Every kind thing from organizing, helping to organize the central nervous system, the neural tube, to organizing limb formation, organs, things like that. So they're extremely important and widely and repeatedly used in development. Same growth factors at different times in development are going to give different effects to the same cell type, because the cells are going to change as they differentiate. They may have transcription factors that they didn't have before and things like that. They can interact with other stuff. So there's a lot of different things that can be changed. It may change different types of receptors that can still bind the same growth factor, all kinds of things. So we can use these things. This is like a basic fundamental toolkit, especially this RAS MAPK pathway, which is so widely used. It's a fundamental toolkit to support embryonic development. And we see this type of stuff, this pathway, used throughout the entire animal kingdom. So clearly it goes back a long time, more complication to get to more higher multicellular eukaryotes, but it's a widely used and very important type of thing. So let's take a look at this pathway. Key point about this pathway: RAS is the name of the G protein involved. MAPKs are classes of kinases. We'll see how that goes in just a moment. Many different growth factors like the EGF family, the fibroblast growth factor family, the hepatocyte growth factors. A lot of different growth factors use these. And of course, there's different receptors for each appropriate growth factor, maybe a couple different subtypes of receptors. Okay, so this plays a big role here. Now, we're going to have a G protein called RAS, but it's a monomeric, not a trimeric G protein. And this G protein does not ever touch the receptor. Instead, it's activated by a series of adapter proteins. Second point about this: once we activate that G protein RAS, it's going to activate a family of kinases, which in turn phosphorylate and activate a second family of kinases, and those activate yet a third and final set of kinases. So we have a three-stage amplification here. So that's another thing. So, in other words, growth factors, because we amplify signals so much, are potent activators and regulators of cell function, even in extremely tiny amounts, like nanomolar quantities or even less, are biologically effective. And in some cases, some growth factors are used clinically for various kinds of things, especially in the stimulating immune system cells and red blood cells. Just think of Lance Armstrong, right? EPO is a growth factor that stimulates, doesn't use this pathway, but it stimulates the proliferation and differentiation of red blood cells. And if you don't get caught, it means you might have a higher oxygen capacity and can cycle harder and longer, or jump farther, or run better and stuff. If you do get caught, you get stripped of your medals in disgrace, and that happens all too often. Yes?
Get a question about the effects of, I know a lot of athletes will go to a higher altitude,
Uh-huh.
train up there, and they're,
Yeah.
they'll have their blood taken out
Mhm.
before a big competition, have it injected back. What's going on?
Okay, well,
at
going
those
up to
guys
higher
make-
altitude, the oxygen is thin, so your body will adapt to a certain point and produce more red blood cells to use what little oxygen you've got. As a matter of fact, case in point, people that are going to do Himalayan mountain climbing, almost always at the kinds of altitudes as, say, Mount Everest or K2, the air is thin enough that only a very few people can actually climb it without the use of supplemental oxygen. It's almost as high as an airplane. And what they do is they spend weeks, and sometimes more, at a base camp at the foothills, which are still like 12,000, 14,000 feet high, and that gives their bodies time to adjust and pump out more red blood cells before they go up. Because if you just go straight, land in an airport and go straight up the Himalayas, you're going to have altitude sickness and it could kill you, literally. Now, the other thing is people that live in very high altitudes. There's Native American tribes around Lake Titicaca out in, I think that's Chile. Well, it's up to 12,000, 13,000 feet up. And these people, if you look at them, and likewise the Sherpas of Nepal will also tend to live quite high up. In fact, they're usually guys that carry all this stuff up to the different camps and Himalayan expeditions. These folks over numerous generations have physiologically adapted to that. They put out much more red blood cells. Their lungs have expanded to take in more air. They're bigger. They often are fairly barrel-chested to hold in the extra large lungs. So there's a number of different physiological adaptations for high altitude and low oxygen levels. Some can occur in a matter of weeks, and that's why people like train in high altitude and stuff. And some it's actually an evolutionary change. But people who have lived for generations up in high altitudes do have significant differences in things related to oxygen uptake and physiology that us ordinary people don't have. That's another reason why a lot of people live in Kenya, the plateau, the Great Rift Valley. That's like, 5,000, 6,000 feet up. And a lot of Kenyans really smoke in the track and field. I mean, we're talking, there are quite a few gold medals won by folks from Kenya because they're used to running at high altitude and stuff like that. They do that all the time, so they're already in really good shape, plus they have altitude adaptations. Now when they come down to sea level or whatever, they kick the tails off of a lot of the competitors. It'd be interesting. I wonder if Usain Bolt actually is native to Jamaica, whether he was born in Africa or where his ancestors came from, because it's possible if his ancestors came from those parts of Africa that he had some of those physiological adaptations as well as just, busting his hump and just, being in superb condition, because he just blew records out of the water a lot of times.
But isn't that anaerobic respiration?
Well, for sprinting, yeah. For long distance running, I guess he was a sprinter. He wasn't through mile, or 1,600 meters and stuff, yeah. But I wonder if that might have helped a little bit anyway. Okay, well, what we got here? Let's take a look at this pathway. We're going to see how this thing works. We start out with a receptor molecule. The receptor binds to whatever the appropriate growth factor is. And there's different receptors for each growth factor, and often many different receptors for a given growth factor. So here's the receptor. Now it binds the growth factor, just call it GF, and that triggers something off. That triggers a tertiary structure change in the receptor itself. And what happens is allows a second copy of that receptor. Now don't forget, membranes are fluid, they're liquid, so proteins can drift around inside the plane of a cell membrane, just like boats can drift around in a lake. Okay, so a second receptor comes in. And it binds to both. Okay, now, that second receptor binding also induces another structural change in both receptors. And it turns out that these receptors are actually protein kinases. In other words, they're enzymes that drop lab manners on the floor. They're enzymes. Okay. They're enzymes that phosphorylate target proteins. In fact, we often call these, and these are unusual kinases because they stick their phosphates on tyrosines, the amino acid tyrosine, and most kinases don't do that. They use the amino acids serine and threonine. And when tyrosine kinases were first discovered, that was quite intriguing. Well, when we talk about another signaling pathway, we'll get to that later on. But, so we say these are tyrosine protein kinases. Now, we activate, this binding activates the kinase activity of both of these receptors. So what do? They phosphorylate each other. That's their target, is their sister. So this one sticks phosphates here, and it's mostly on this tail end that sticks deep into the cytoplasm, and we stick phosphates over here. So everybody's sticking phosphates on each other. So the receptors phosphorylate each other. Okay, now what does that do? Turns out that actually... yes.
Okay, so the receptors are the kinases.
Yes, the receptors are actually kinases. They don't activate unless they stick together bound to a growth factor. If they're on their own or there's no growth factor present, at least that's what it's supposed to work, the receptors won't phosphorylate each other. Now there are some mutations, and we'll see this later, in certain of these receptors where they will bind and phosphorylate each other even when there's no growth factor present. That is a bad thing because this pathway stimulates cell division and differentiation.
So that would like cancer?
Exactly. In fact, there is a drug that blocks that. It's called Receptor. It's widely used in certain types of cancer if you have the appropriate mutation. In fact, Dr. Romano was part of Dr. Romano's treatment last year. Unfortunately, it didn't really work, but they tried. He did test positive for that receptor mutation, so they were using that. So that's actually widely, it's been out for a number of years, and it blocks those kinds of receptors so they don't phosphorylate each other.
So, what kind of cancer do you have? Is that?
Oh no, I've got a type of leukemia, and that uses different signaling pathways. Yeah, the main problem I have is I'm missing a piece of one of the chromosomes that happens to have this gene called RB, which is a known oncogene. It stimulates cell growth when it's improperly active. And I'm missing something called P53, which is common in a lot of cancers. And what that does is it detects DNA damage before the cell divides, and the DNA is damaged. It says, halt the countdown, don't divide, we got to fix this mess. And calls out the repair enzymes. And if it's irreparable damage, then it activates a second pathway called apoptosis, and that kills the cell.
Okay.
Yeah.
So
I got a double whammy there. And I'm also missing a microRNA that also ramps down cell growth. So I'm getting getting 3 ways it runs here. Yeah, I was getting somebody say, I'm going to lose any 2 genes, these would be at the bottom of the list, you know. Okay, so that's a thing. Pointing around at this graduate seminar I do in the fall, I might try to do something in the cell molecular biology of cancer. Not so much the clinical stuff, leave that to the docs, but you know, what goes wrong with cells when they do that? Because it's interesting for all the pre-med types. Plus, it's biologically quite intriguing. I just don't advise getting it yourself. You know? Okay, yeah, I think I would like to miss that step, but too late. Okay, well, at any rate, what do we have here? We've got these phosphorylated receptors, right? Okay, it turns out that these phosphates here are recognized by a family of adapter proteins. They recognize and bind to these phosphates, and they don't bind to the unphosphorylated receptor. And these proteins fall, they're often called the SH2 family proteins. Okay, and SH2 just stands for certain structural feature. These things can do a number of different things. Some of them can activate parts of other signaling pathways, but the one we're mostly interested in is going to bind to and alter the structure of a guanine nucleotide exchange factor, or GEF. Now these GEFs, once again, are regulatory proteins for G proteins. They bind the appropriate target G protein. And what they do is they stimulate that G protein to get rid of the GDP and pick up a GTP. In other words, they tend to activate the G proteins. Now, the pathways we're looking at previously, it was the receptor that directly did that to a trimeric G protein. In other words, the receptor was acting as a GEF. Here we don't have the G protein ever even touches the receptor. Okay, now, the G protein in question is a protein called RAS. It's a monomeric G protein. Now, I'm drawing this, that squiggly line represents that RAS has a covalently bound lipid to part of its structure, and that lipid acts as an anchor to get the thing into the cell membrane. It anchors in the cell membrane. The protein itself, the surface of this protein is quite polar, but that lipid anchor holds it in the membrane. So it's just underneath the membrane. So what's going to happen is it stimulates, the GEF stimulates this GDP out and GTP in. God, shut up. I gotta watch it. I'm gonna slip one day and describe this chocolate, so I'm gonna go up on YouTube. And have a few expletives here, old Anglo-Saxon expletives. Okay. All right. Okay, now, once RAS is bound GTP, once we've done that exchange, RAS undergoes a radical change in tertiary structure. Now we're in the active version of it. So I'm going to represent that as a square. Okay, active RAS can bind to, what's up?
I don't know. It's just biology. We're all crazy.
God, I thought you had to get a PhD to get that loopy.
It's Sauterer loop.
Oh, okay.
Started
Premature mental degradation, right?
Well, me and her had organic class.
Okay. So you guys are sniffing all the aromatic solvents, right? Oh, I love toluene, man.
No.
Okay.
It was benzocaine.
Okay, that'll do it.
I don't read the bottle.
Those
They
last
finally
week's
got the hoods working.
Excuse me?
They finally got the hoods working.
Oh, okay.
Molly, this
All right, well, maybe
is
you
the
guys are...
first lab we have ever had the hoods working.
Jeez. Oh, great. You
I
work
mean,
with
so we
flammables,
might actually
corrosive,
So when we
toxic...
get cancer, we can blame it on
Oh, boy,
GST.
okay, yeah, okay, so you guys are gonna follow in my footsteps if anybody comes down with leukemia next year. I'll recommend a good oncologist.
We'll call you.
Okay, all right. Okay, well, anyway, okay, we've got RAS activated. Once again, active G proteins, they bind to and regulate other types of proteins.
I'm about to take a picture of it.
Okay, as long as you're not texting your friends, just your Facebook. Okay.
No, I'm not.
Study tool.
Well, all right,
There you
that's
study.
fine, but so many people, especially in the freshman classes, you know, all they're doing is looking at Facebook, posting stuff on
Pinterest.
Facebook, and texting their friends in the middle class. I see people do it when they're walking across the street, and then they're gonna sue the daylights out of you if you run them over because they didn't know you were there, you know? Okay, that's actually happened. I get a few people get hit every year on
I
this
was in the
campus.
crosswalk and they didn't stop until I finally was clean.
Yeah, and a lot of it's people.
Big
just
old
not
bug
paying
eyes.
attention.
You good?
Mhm.
Oh,
Oh, yeah.
thanks.
Okay, do you have your lab, Ryder?
I have to go print downstairs, she's not in there.
All right. Okay, all right, so we have active rest. Geez, I'm going to you all. Guard shooter. Okay. Oh, all right, go. No, I don't want to, I have to clean up the mess. Okay. That would be good. That would be very bad. Okay, that's the biochemistry here. All right, active RAS. It binds to and activates a set of protein kinases. And here's our first step of amplification. These kinases are called the RAF proteins. There's a couple different versions of them, but they're a set of protein kinases. Once they're activated, we go to our second step, our second amplification step. And that is a second family of kinases that get phosphorylated by RAF. And they're called, abbreviated, the MAPKs, they're kinases. And we'll see what that means in a moment. Okay, these guys, our second step is activated. Now, we go and phosphorylate and activate our third and final set of kinases. Everywhere. Okay, and that's what we call the MAPKs. And that's a third set of kinases. And they get phosphorylated by the second set. Okay, once we've activated this final set of kinases, what happens is these go out and phosphorylate all kinds of things. They can phosphorylate numerous transcription factors and proteins involved in chromatin structure. Of course, that's going to stimulate changes in gene expression, turning genes on or off. They stimulate proteins that ultimately are going to activate the cyclins and CDKs that are the kinases that fire up the different parts of the cell cycle, including mitosis. So in other words, they stimulate cell division and or cell differentiation, depending on the cell type. And the reason that the same pathway can activate different things in different cells is because, once again, there are other types of transcription factors in cells that will interact with the ones you've activated and choose what genes are going to be regulated, how much they're going to be regulated. Also, there are different members of these different families. Like, when you think about 3 or 4 different versions of RAS, I think there's several versions of these guys, and there's a whole slew of versions of these. If you take biochemistry, Dr. Nichols will give these guys names. I'm just giving the family name and stuff. But common ones have specific names. I think ERB2. Has anybody taken biochem here? Okay. If you hear ERB2, we're talking about one of these guys. Good. Okay. Now, that's the way the pathway works. Okay. Widely used in all kinds of developmental events. But, not surprisingly, there's a lot of things that could potentially go wrong with this pathway. And when it does, that means it can start stimulating cell division when you don't want that to happen. In other words, this pathway is involved in numerous human cancers. Not the sole cause by any stretch of the imagination, but it plays important roles. Maybe 40 or 50% of human cancers have defects in this pathway, improper activation, and things like that. So what we're going to do now is we are going to take a look